The title of my talk is Microstructure and Rheology of Cellular Blood Flow. And then because of the really nice and motivating discussion that we had yesterday in the margination subgroup, I quickly changed the presentation so that I still cover rheology, but very briefly, and then I'll actually put in a lot more slides on platelet margination, and I'm going to talk mostly about our results in terms of platelet margination, because I think that's really where the interest of this group is. So I want to thank, first of all, former and current students. Daniel Rieser and Jonathan Clausen are former students who did a lot of the development of the computational method and analysis, both in terms of blood rheology and margination. And Marmar Mehrabadi is the current student who's doing both computations and experiments with my colleague David Koo, who is mostly on the experimental side. So just to motivate the talk, what you saw here happen is that in the few minutes that I was doing the introduction of my talk, we saw that this model of a stenosis, which is collagen-coated glass tube, essentially clogged in the few minutes that we were discussing. And this is really one of the reasons we are interested in blood rheology, in platelet margination, because when there is a rupture in occlusion, it only takes a matter of minutes. It's a very short time before these platelets accumulate and there is a clot, complete stenosis formation and complete occlusion. So if we take the availability of platelets and the regular diffusion of platelets into the boundaries, we can never get the number of platelets that accumulate in this very short time. So one scenario, one idea is that, okay, this platelet margination really plays a big role, and this RBC interaction with platelets play a big role in this. So this is one of the motivations for looking at blood rheology and platelet margination. And by the way, these two are related, so hopefully I can convince you that rheology and platelet margination are related. So just very quickly, I'll very quickly cover the computational approach that we take. The fluid part is solved with the Lattice Boltzmann method. If you're familiar with this method, you know what it is. If you're not, it is a particle-based method. It's based on the discrete Boltzmann equation, but then the equilibrium distribution is such that it mimics the Navier-Stokes equation, and it really gives the same solution as a Navier-Stokes solution for incompressible flows, although Lattice Boltzmann is pseudo-incompressible. The fluid-solid coupling is a full two-way coupling between the fluid and the solid. And this method started in our original paper in '98, where we developed this coupling between fluid and solid for direct simulation of particles suspended in fluid. And then the RBC membrane, we are using the Spectrumlink method for the deformation of the membrane. As I said, Lattice Boltzmann is a particle-based method. If you want to know more about it, in 2010 we had an annual review article describing Lattice Boltzmann and its progress to complex flows. Essentially, we can recover the continuum variables from the distribution function, the particle distribution functions, based on this quadratic equilibrium distribution function. The Spectrumlink modeling for the membrane, we essentially follow the method developed by these investigators here. And then our contribution was to take this and then couple it to the Lattice Boltzmann method. So that's the coupling between Spectrumlink and Lattice Boltzmann. And for those of you who want to learn more about this coupling, fluid-solid coupling, we have a series of papers here that describe in detail how this coupling is done. And by the way, there is a lot of validations for this computational method. So I'm not going to go through the validation. So we should talk about the dimensionless parameters that are important in this analysis. So we define a capillary number, which is based on the membrane shear modulus. And so it's a ratio of the shear to the shear modulus of the membrane. Then the particle Reynolds number, so that's based on shear rate and the radius of particles. So it's this is what determines the importance of the particle inertia. And then also when we are doing tube flow, the tube Reynolds number, which may or may not be important. So again, a number of validations that we have gone through, and we are convinced now that the simulations do capture the formation of the red blood cell, which is critical in these analyses. Also, the dynamics of red blood cell is important, and we have done a series of benchmark analysis showing that we do capture the physics, the dynamics of red blood cell movement, both in terms of tumbling and tank threading. Also, this parachuting, there have been a lot of experiments in the aspect ratio change in these parachuting shapes, and we have done comparisons of those as well. One important aspect when you're doing suspension is to be able to capture the particle-particle interaction. That's really very important when we're doing dense suspension. And we use a Morse potential type model to mimic the cell-cell interaction. And we can resolve the interaction as compared to experiments to very, very small distances in the scale of nanometers. So we are comfortable with doing dense suspensions and analyzing the particle-particle interaction. When we are studying rheology, we want to get rid of Wall effects. So in order to get rid of Wall effects, we use this Galilean shift or the Lees-Edwards boundary condition, where essentially there is a Galilean shift in the computational cell or domain that we have up and down in order to preserve the shear, but to get rid of the wall. So when I talk about rheology, we don't have a wall to impact the rheological parameters that we get. And this is really important in any kind of rheological analysis. So how do we get the rheological parameters such as relative viscosity, normal stress difference, and importantly, the particle, the suspension pressure? So we have the fluid stress tensor and the contribution of the stress tensor due to presence of the red blood cells. And then we use this stresslet, which is essentially an integral over all the traction over all of the particles. So this gives a true stress in the suspension. And from this, we get the total stress tensor. And of course, once we have the total stress tensor of the suspension, then we can get the relative viscosity from the shear stress, and then the two primary and secondary normal stress differences. Okay? So this is just a quick background. Now, we find the same shear thinning effect due to the same shear thinning effect in relative viscosity that others have found as well. And what we can show is that this effect of shear thinning has two main causes. One is the deformation of the red blood cells. And as you can see, I think you can see that in the higher shear rate or higher capillary number, the red blood cells are more elongated. And also in terms of the average angle of the red blood cells as they form in this shear layer. So for the same range of capillary number, if we look at the average angle of the red blood cell compared to the shear layer, we see that as we increase the capillary number, the angle decreases, the mean angle decreases, and this is also contributes to lower shear in the suspension. It's important to also look at the normal stress differences, or here we are showing the three components of the normal stress, and you see that as the capillary number goes to 0, we are essentially saying that the particle is not deforming. Okay, so you see that the primary normal stress, which is in the direction of the flow, so 1 is the direction of the flow and 2 is normal to the flow in the shear gradient direction, the primary normal stress actually changes sign. And this is because of the deformability, the elasticity that's present in the red blood cells. So this is important in terms of the particle pressure. Because of this change in sign, the particle pressure also changes sign as capillary number is increased. So it goes from positive, which is compression, into negative, which is suction. So this can explain, so this relates to what we observe later on that I show in terms of the migration of the red blood cells in a tube flow. So as I said, I'm not going to spend too much time on the rheology because I think there is more interest in the margination, but it's important to consider that this elasticity and the suspension in the suspension plays really a very significant role in terms of the macroscopic rheological parameters. In terms of the microstructure of the flow, it is really important to consider that as the red blood cells are moving even in a tube, not in a shear layer, so now we are shifting from a shear layer into a tube flow, there is continuous formation of clusters in the compressional direction and then breakup of clusters in the extensional direction. So even though this is not a shear layer where there is a very distinct, well-known compression and extensional axes, we do still repeatedly get this formation and breakup of the clusters, which has a big effect in terms of the red blood cell rheology. So what is the consequence of this? So now if we include platelets along with red blood cells and then look at the interaction of these two, and we model we take different shape of platelets, but normally we model platelets at oblate spheroids, and these are the average sizes of a platelet and a red blood cell. So now if we have only platelets in a tube flow, as we show here, so this is only platelet in single phase flow, and the platelets have slight amount of inertia. So there is, in my view, there is always slight amount of particle inertia. For very, very long time, what happens to where do the platelets marginate to? Well, it's been known since early 60s that in a situation like this, there is a band called the Segre-Silberberg band where particles marginate to that band, which in this case is R over R is 0.6. And this is partially verification that our code can handle the, this margination of particles. But now if we include red blood cells and take different shapes of platelets. So in this case, we are taking spherical platelets. Here, the actual oblate spheroid platelets, and here, disc shape. I wanted to notice a couple of things. One is, as soon as I started this animation, the red blood cells were at the center. But the platelets are still being bumped around with the red blood cells, and they're gradually moving outward and getting trapped in this what we call skimming layer or RBC depleted layer. Now, if you look at the the actual platelet-shaped particles, it's essentially the same. Immediately, the red blood cells go toward the center, and the interaction of the platelets with the red blood cells is just bumping around the platelets, and they are trapped in the depleted zone. You'll see the same for the disc-shaped particles as well. In fact, if we let move long enough, we show that almost all of the platelets go towards the skimming layer. Once in a while, there's a platelet that could move back in, but then it gets bumped around and goes in. The platelets that are at the center take much longer time to move out because the center is a point of equilibrium and very low shear. So, and here we show that as we increase the aspect ratio, the rate of margination decreases. So in my view, if we want to analyze the physics of margination, we really should also look at the rate of margination based on different parameters. Yeah.
So you're saying irrespective of the size and shape, they're all marginal?
Yes, there is always margination.
Like 90%
Well, there are some that come in and go out, so it's not a complete 100% because it's a diffusive process, but eventually most of them marginate outward. So, okay, so now if we look at different parameters, the effect of different parameters on margination. So as we increase the hematocrit from 20% to 40%, the margination rate increases. Lambda is the ratio of the viscosity of hemoglobin to the liquid outside. And if we go from 0.5 to 0.5, which is the actual physiological ratio, and much higher, we see that the rate of margination decreases, and this is the effect of the aspect ratio. So now let's look at the cause of platelet margination. So if we take a test case where we put a few rigid RBCs, or we put a few small soft RBCs or small rigid RBCs into regular RBCs. So these are like little computational experiments. So we have 3 different cases, and this is in a channel flow of 40 micron, hematocrit of 20%, and lambda is 5. We show that, okay, so this is 40 micron. The red is the amount that the soft RBCs migrated to center. So we take that amount and we just add it to the center, which is at 20 micron. Then the other 3 are rigid, small soft, and small rigid. And at least to our surprise, we saw that small rigid RBCs are migrating faster than the other 2 cases. So it is not only the size or the softness and rigidity, but it is both the size and the the rigidity of the platelets that is contributing to higher rate of margination. But how about shear? What is the effect of shear rate on margination? So we have two cases where we change we have a case where we change the shear rate from 1,000 to 20,000 reciprocal second. We look at the margination of the RBC and the platelets in this channel versus time. Of course, as the shear rate increases, the margination is much, much faster in terms of time. But if we scale it to distance, you see that the curves essentially collapse. And shear rate has some effect, but it's not a significant effect. It's not the dominant parameter in this. So, okay. So now, what would be the effect of the channel size on platelet margination? So we are taking now 3 different channel sizes, 40, 80, and 160 micron, and looking at the platelet margination. So the larger the channel, the slower the rate of margination with respect to the displacement. However, let's scale this. So if we take this, if we do the margination length scale based on diffusive length scale, so interaction with the RBC and diffusion due to this interaction, and we take y, so the lateral direction to be scaled as the diffusion coefficient d. So the time to steady state scales as h squared over 4d. And then if we take the shear-induced diffusion to be a function of hematocrit times shear rate times a squared, this is typical in shear-induced diffusion, then the length scale for margination, taking the average shear rate to be 3 times velocity over the channel height, the length scale of diffusion would scale as the channel height cube over a squared, the radius squared. So now if we go back to this data that we have and we scale the length according to this length scale, h cubed over a, which I've done here, you see that all of the curves collapse. So this demonstrates that this diffusive length scale is a real good scale for the margination. So the hypothesis that has been around that it is the shear-induced diffusion that causes margination, and what I refer to as the RBC-enhanced shear-induced diffusion seems to be the cause of margination. So now if we know the mechanism of this margination, maybe we can model this. Let me move on to the model. So we take a general convective diffusive model for margination, but we add what we call a sink boundary condition for this equation. So this is just the regular convective diffusive equation. P is the concentration of the platelets above the RBCs, but we see that the concentration in the depleted zone is just 0. So as it goes in the depleted zone, it just disappears. Okay, so sorry, the P is the platelet concentration, not the RBC. So in this analysis, we need 2 pieces of information. We need to know the size of the depleted zone delta, and we need to know the diffusion coefficient, which is a function of y. But the nice thing about computational simulation is that we have both of these. So we can measure the size of delta, and we can take the movements of the platelets, and we can calculate the diffusion coefficient locally. So we get the diffusion coefficient locally as a function of y. And if we plot this for different channels. Of course, in the depleted zone, it should be 0. Then it increases very rapidly, so the maximum is very close to the depleted zone around the boundaries. And then it goes back. And as you can see, when the channel size becomes much larger than the RBC diameter, as it should be, the diffusion coefficient goes to 0 at the center. Okay, so theoretically, if the channel size is much, much larger than the RBCs, shear rate at 0, at center is 0, so the diffusion coefficient should be 0. Now, then we compare this model. So these are results that we have for the model with different sizes of the depletion zone, but the green is the actual size. And we see a pretty good agreement with experiments, except we are over-predicting the concentration of the RBCs in the depleted zone. And I'll explain why that is. So if we plot the comparison of the direct simulation to the model, we are getting pretty good agreement for between the two. So the reason that we are over-predicting the concentration in the depleted zone is that we are assuming the depleted zone is an absolute sink. So anything that goes there stays there forever, where in reality the depleted zone is a diffusive sink. So once in a while, actually, the platelets do go back and forth between, and those trajectories that I went over quickly actually showed that some platelets actually go back in. So there is some diffusive effect there. But in general, this model, I think, in terms of demonstrating the physics of this process I think it gives some, represents the physics of what is going on. So just very quickly to summarize, I know I'm out of time. We see that, so a couple of things. You can read the summary, but a couple of things that I want to emphasize is that rheology, particle pressure, plays an important role in explaining why the RBCs are accumulating toward the center. The physics of margination, we believe, is what I call the RBC-enhanced self-diffusion. So it is really the interaction of the platelets with the RBCs and in conjunction with the depleted zone where this migration occurs. So both are important. And keep in mind that the diffusion coefficient is a function of radial direction. So you cannot take a constant diffusion coefficient and say that, OK, this would predict margination. It won't. Because as we saw, diffusion changes greatly from the center of the tube to the boundary, and it drops 0 to the skimming layer. So with that, I'll stop. Okay.
Thank you very much.